Hi, I didn't see you there. I was just reading my ecology textbook. Do you know what some interesting concepts are? Seed dispersal and mutualistic relationships. I wonder if there's any literature published on this idea. Oh, would you look at that? New mutualism for the old. Indirect disruption and direct facilitation of seed dispersal following Argentine ant invasion. Oh, I better go show the guys. Hey guys! You wouldn't believe it, guys! Look at this nifty paper. You'll never guess what it theorizes. It hypothesizes that the invasion of Argentine ants would disrupt the mutualism between native ants and plant species. They theorize Argentine ants would fail to substitute for the seed dispersals completed by native ants, and also that the ants would favour seeds of smaller size and reward, changing the dynamic of seed dispersal.
Gee Dan, that sure is interesting, but what is mutualism? I've never heard of it before.
And I've never heard of an Argentine ant before. They don't sound native.
And those sure are some interesting hypotheses. They've proven to be correct by the use of experimental data collection and statistical analysis.
Why tell you when I can show you? Let's go.
Oh boy, an Argentine ant nest. I can't wait to meet them.
Let's have a little look closer.
The Argentine ant Myrmecochory humile is one of many species that form the family of Firmicidae. Derived from the order Hymenoptera, they share early lineages with bees and wasps. L. humile is on average 2 to 3 millimeters long and is medium to dark brown in color. Generally, the body of the Argentine ant is smooth and shiny. However, they lack in stingers, a common attribute amongst ant species. The morphology of the workers of L. humile are identical, and this is a contributing factor to nest sharing of worker ants, resulting in nearby nests forming supercolonies. L. humile is a native to northern Argentina, Brazil, Uruguay, and Paraguay. Their preferred environment are regions with moderate temperatures and moist climates. However, the Argentine ant can be found in a variety of natural and anthropogenic affected areas. Recorded sites include agricultural, natural forests, and urban environments. The climate is also very varietal, with the ants having been recorded in Central and Southern North America, Hawaii, Japan, Spain, and Southeast Australia, proving that they are a very successful invasive species. The diet of Argentine ants consists mainly of sweet plant material, such as the lysosomes and nectar. However, the ants have a taste for blood, being known to feast on other slow-moving insects, their larvae, and eggs.
In botany, a diaspora is a general term for a dispersal container consisting of a spore or seed, paired with additional tissue that assists in its dispersal. The tissue is most commonly an edible part of the seed that promotes transport or consumption by another organism. An example of this is the seed dispersal interactions by ants, termed Myrmecochory. When plants that take advantage of this method of dispersal produce a seed diaspore within a lysosome as a food source that is attractive to the ants. The lysosome encourages the transport of the seed, with the food source often being transported back to the ant colony. This mutualistic relationship provides the ants with the source of food and the plant with a reliable method of dispersal.
The first experiment focuses on the impact of ants on seed dispersal. For this, 2 site types were monitored. They included 3 sites with a present population of Argentine ants referred to as the invaded sites, and 3 sites without Argentine ants referred to as the uninvaded sites. Argentine ant presence was confirmed through baiting, trapping, and identification.
Honey. Tuna.
The experiment focuses on seed dispersal of 4 plant species, 2 native and 2 invasive. Acacia sophorae and Acacia retinodes represent native species, while Polygala myrtifolia and Dipigon lignosus represent the invasives. All seeds used in the experiment were collected prior to the study from the Mornington Peninsula National Park. Each seed varied in mass and elaiosome size. This means variability in work and reward ratios.
Prior to pinpointing the presence of Argentine ants at the various locations, the 6 sites were established. The sites were scattered along 4 kilometres of the park boundary with an average of 750 metres between the sites. It is important to note that the boundary of the coastal shrubland sites were adjacent to peri-urban environments. However, the vegetation did not vary greatly across the uninvaded and invaded sites. A 10 by 6 plot consisting of 24 grid locations was created, and each grid was spaced at 2 metre intervals across the 6 sites. Within the grid, random allocations of tray and mesh combinations were laid out. There were 3 replicate trays for each seed combination, resulting in 12 trays for each 10 by 6 plot. Each tray was made from perspex that was lightly sanded and measured 6 by 6 centimetres. The tray was surrounded by mesh and a cage with dimensions of 15 by 15 by 8 centimetres. This cage was also topped with clear plastic film. The mesh, cage and plastic film were used to limit interactions and disturbance from other fauna and abiotic features like wind and rain. Initially, 10 seeds were placed on each tray. 30 seeds for each plant species was used across the plot with an additional 3 replicate trays, meaning in total each 10 by 6 plot contained 12 randomly assigned trays with 120 seeds overall. Seeds were then replenished at 24 and 48 hours to maintain availability. Finally, a tally of the seeds removed was taken after 72 hours.
2 aspects of seed dispersal were examined separately at one of the 3 invaded sites. These aspects were distance of seed dispersal and rate of seed burial. Seeds of Acacia retinodes and Polygala myrtifolia were marked with coloured paint to assist in tracking seed distance from the tray. Again, 3 replicate trays for each seed species were established, each containing 10 seeds. The seeds underwent an application of paint proximal to the elaiosome attachment. Trays were placed at random grid locations across the 10 by 6 grid with at least 2 metres separation between the trays. Factors considered in this part of the experiment include the time between the seed being discovered and removed, the number of ants assisting in the removal of the seed, the distance that the seed is moved and the final position where the ants leave the seed. Soil cores, 10 cm long and 4 cm in diameter, were taken from the entrances of 5 Argentine ant nests and 5 randomly selected areas away from the nest, in order to look at seed burial rates. These were sieved so that seeds could be extracted, identified and counted.
The second experiment conducted was to replicate seed removal based on seed reward ratios. For this, artificial diaspores were created. The purpose of creating artificial diaspores was to control the examined effect that seed mass and elaiosome size has on dispersal by native ants and the study subject, the Argentine ant. This was done at a site adjacent to the Mornington Peninsula National Park. Collection of seeds from Acacia retinodes was done in order to extract individual lysomes from the seeds. The lysomes were then adhered to the various sized beads to create the artificial diaspores. There were 2 bead sizes used for the artificial seeds. They were 3mm and 4mm in diameter. Elaisomes varied in size also. Sizes of a lyosome were 5 and 20% of the total mass of the bead. This was used to simulate different reward ratios. The reward ratios chosen in this study represent real-life ratios from averages of the species Polygala myrtifolia and Acacia sopherae. Beads without elaisomes were included as controls. There were 5 seeds of each bead type placed randomly at different points across a 12 by 10 meter grid with 2 meter boundaries between trays. Each tray consisted of 5 times 3mm beads and 5 times 4mm beads with 0, 5 and 20% reward. Additionally, 10 extra diaspores from Acacia sopherae and Polygala myrtifolia were added to each tray. In total, the trays consisted of 40 seeds. This was consistent across each 2 by 2 meter grid site. A count of species of ant present and frequency, seeds that were moved greater than 5 centimeters, seed burial distance, activity upon seed lyosomes, and the seeds that were not moved, with additional counts recorded at 9 p.m., 1 a.m., and 6 a.m. to help understand the nocturnal interactions of ants.
Time to hit the lab. Let's go!
Lastly, statistical analysis was done in order to draw conclusions from the gathered data. Data was transformed when necessary to abide by Systat-10's software capabilities. Firstly, a partly nested two-factor ANOVA was performed in order to determine how Argentine ants' presence affected seed species and seed dispersal. A two-factor ANOVA was also done on individual sites, testing the effect of individual seed species. Subsequently, Tukey's Honest Significance Difference Test, or a HSD test, was then used to confirm the dispersal differences across the seed species. The invasion effect of each seed was tested separately across the sites. Two-factor ANOVAs and one-factor ANOVAs were used to examine the effect of percentage reward and seed size on removal, and to make comparisons between real seeds and artificial seeds. Subsequently, Tukey's post hoc test was used to confirm dispersal differences due to reward ratios across the seed species and artificial seeds. Data then confirmed significant were modeled appropriately with two-factor ANOVA results and hypothesis testing. One-factor ANOVA testing was corrected using degrees of freedom and mean square error bars within models.